Hey, so our next talk is by Ambuj Upadhyay from the University of Minnesota, and his title of his talk is Muscle-Derived TGF-beta Growth Factor Myoglianin Regulates Size of Imaginal Wing Discs.
Thank you to the organizers for giving me this opportunity to present my work here at this unique conference. My name is Ambuj. I'm a grad student in Michael O'Connor's lab. Let me see if I can use this mouse. Okay. And today I would like to share with you my ongoing work on the role of TGF-beta signaling in regulating imaginal discs. But before I move on, I just want everybody to know that I will submit this image for next year's Image Award. Hopefully I win. I wish I had a Correlio GFP right there in Florida just to mark Orlando, but ongoing work. Okay. So, as many of you know, the TGF-beta superfamily can be divided into two subfamilies based on sequence similarities. These two families are the BMPs and the TGF-beta activin. Now, the core signaling components are widely conserved in metazoans, and Jennifer earlier talked about the signaling pathway, but briefly, I would like to go over that again. Signaling is initiated when the peptide ligands bind to two type 2 and two type 1 receptors on the plasma membrane. And this binding activates the serine-threonine receptor kinases, which in turn phosphorylate the cytoplasmic transcription factor SMADs. The activated SMADs then localize to the nucleus and with cofactors either activate or repress target genes. Now we know a lot about the role of the BMPs during wing disc growth in Drosophila, but so in Drosophila, the ligand DPP, homologous to the vertebrate BMPs, acts like a classic morphogen and to regulate both patterning and proliferation. These two processes occurs during the larval growth period, which is when defects in signaling can result in improper tissue growth. As an example, showing you here in the bottom, as a gain of function in the DPP pathway leads to ectopic lateral growth along the anterior-posterior axis here, as well as defects in wing vein positioning, which I'm not showing you here. So on the contrary, we know very little about how the activin pathways contribute to the organ size. But today I hope to convince you that the TGF-beta ligand myoglianin expressed in the body wall muscle acts on the imaginal discs to promote tissue growth. Oops. So my story begins when our lab made the key observation that the loss of different genes in the activin pathway leads to dramatically different wing size phenotypes. So loss of baboon, the type 1 receptor, which is the ligand binding type 1 receptor, leads to a smaller wing size versus loss of DSMAD2, shown in the bottom left, leads to a very large wing disc size, notably with the ectopic lateral growth here. The second surprise came when we discovered that baboon is epistatic to DSMAD2. That is, that baboon and DSMAD2 double mutants look just like baboon mutants. This suggested to us that the reason for the DSMAD2 phenotype here is due to the presence and activity of baboon. Furthermore, because the DSMAD2 mutant phenotype phenocopied the BMP gain of function mutants, we decided to check the activity of BMP pathway in the DSMAD2 mutant discs, and which can be done using a Brinker, that is incorrect, which can be done using the Brinker-Lag-Z reporter. Now, Brinker is silenced by DPP in the middle of the disc, resulting in the pattern as you can see here. This figure should be on this side here. So, oh, there it is. Okay, so you can see the pattern of the wild-type Brinker reporter, which is in green here, which is expressed in the flanks of the disc. And when we looked at this reporter, when we checked the reporter expression in the DSMAD2 mutants, we saw it was completely silenced. So from this observation, and along with others I'm not showing you here, led us to formulate a model in which DSMAD2 is behaving like an anti-SMAD and inhibiting ectopic baboon activity. In the absence of DSMAD2, this purple protein here, baboon is uninhibited to ectopically silence brinker, particularly by cross-talking with the BMP pathway components such as MAD, Medea, and Schnurri to silence brinker. So this led to a series of brand new questions to answer. If baboon is activated, then which isoform of baboon is required? There are 3, which I'll show in the next slide, and which type 2 receptors are required in the disc. And then what ligand is corresponding to these receptors? And where is the ligand coming from? So our first goal was to identify which of the 3 splice isoforms of baboon which differ in their ligand binding domain, and which type 2 receptor is important for DSMAD2 phenotype. To do this, we made RNAi clones in the wing disc and assayed for brinker silencing in the clones. So when we make a DSMAD2 RNAi clone, which is illustrated by the top panel here, the RNAi is expressed in the clones marked with GFP. You can see that in these clones that the brinker reporter is ectopically silenced compared to the wild-type neighboring cells. And we see this as both here and here as holes in the native pattern of the reporter. So subsequently, when we generate RNAi clones for DSMAD2 plus baboon A or DSMAD2 plus PUNT, the type 2 receptor, we see that the brinker reporter is restored in these clones. This means that baboon A and PUNT are required for the ectopic brinker silencing that you see in these DSMAD2 RNAi clones. I did the double RNAi clones of DSMAD2 and WIT, which is the other type 2 receptor that could play a role in the disc, and the other 2 splice isoforms, but they did not show any difference compared to the DSMAD2 alone. So we reasoned that they're not required for silencing, and only baboon A and PUNT are the active receptors. And now, so others have suggested that all of the isoforms could be functional in the disc, but this assay suggested to us that only baboon A is functional. And when we checked by qPCR which receptor isoforms are available in the disc or are expressed, we only detected baboon A, further validating our initial results. So we next asked the question, which activin-like ligands is functioning in the disc, and we reasoned that since baboon is epistatic to DSMAD2, there must be a corresponding ligand that is also epistatic to DSMAD2. And because we saw evidence of baboon A being functional, we initially thought that perhaps myoglianin would be epistatic, but we wanted to independently check all 3 ligands. So when we did this experiment, we discovered that only myoglianin is epistatic to DSMAD2 and rescues the DSMAD2 phenotype. You can see this is the mutant phenotype, and this is the double mutant phenotype. The double mutants of DSMAD2 in activin or DSMAD2 in dawdle do not show any difference compared to the single mutant DSMAD2. So this again suggests to us that myoglianin plus in combination with baboon A and PUNT work together to regulate tissue growth of the wing disc. Now this growth control of myoglianin or by myoglianin is a novel finding, and we asked whether this was specific to the DSMAD2 mutants or does it occur in the wild type animals as well. Now to answer this question, we compared the size of the 3 discs that are often found together, the wing haltere and the third leg disc, of both myomutants and the heterozygous controls. And when we did this, we discovered that the loss of myo resulted in smaller imaginal discs, all 3 discs. But the ratios, which is seen in the red here in the top figure, but the ratios of the discs do not change, which suggested to us that myoglianin acts on these tissues equally. We next asked the question, where is myoglianin secreted from? The gene myoglianin gets its name from old in situ data showing expression only in skeletal muscles and glial cells, so hence myoglianin. And others have suggested that maybe, perhaps that the wing discs are expressing myoglianin as well. So we wanted to test whether the disc could be producing its own myo or it's getting it, or the signal is coming from the muscle or the glia to produce this kind of mutant phenotype. So we first checked the expression pattern of myo by taking its promoter region and placing it upstream of GAL4 gene. We generated the fly line and crossed it to a US CD8 GFP. We observed that the GAL4 was expressed solely in the larval body wall muscle and the glial cells in the larval brain. It's weak, but you can see it's expressed there. But it's not expressed in the disc cells, which are right there. And with this information in mind, we wanted to functionally test where is the source of myo that is acting on the disc, either the larval brain or the skeletal muscle. Now to do this, we performed a genetic epistasis analysis with a slight twist. We reasoned that in the DSMAT2 null background, if we remove myo expression using RNAi in the relevant tissues, then it would rescue the DSMAT2 phenotype. So we did the necessary control by ubiquitously knocking down myo using the daughterless GAL4 line, and we showed that we could rescue the overgrowth phenotype seen in the mutants. And this validates the RNAi, or it shows that RNAi is functional, and it also phenocopies the DSMAT2 myo double mutants. So with this in hand, we were confident that we could do the tissue-specific RNAi and test which tissues is the relevant tissue that myo is secreted from. So here's the punchline. When we performed the tissue-specific RNAi of myo, much to our surprise, only knocking down myo in the body wall muscle using two different drivers, MEF2 and MHC, rescued the DSMAT2 phenotype, but we did not observe any change in the tissue size when we knocked down in the glial cells using RepoGal4 or the wing disc using escargot-Gal4. So we can also demonstrate a rescue experiment by resupplying myoglianin in the muscle of myo mutants, which rescues the wings disc size phenotype. So again, I showed you earlier that in the mutants, the discs are smaller, and we can rescue this small size by overexpression of myo in the MEF2, in the muscle using a MEF2 driver. So to summarize, our data suggests that during larval growth, myo is secreted from the body wall muscle, and probably it goes into the hemolymph and acts on the wing discs to stimulate growth to wild type levels here. However, if this growth signal is not restrained by DSMAT2, it can lead to tissue overgrowth due to hyperactivity of baboon, the activated receptor. So in conclusion, this work highlights a novel role of myoglianin in Drosophila, where perhaps there's speculation that it's used to coordinate scaling between appendages and the muscles that powers them. It's also noteworthy that the larval wing disc can affect the size of flight muscles as well by wind signaling. So now these two forces of wind signaling and myoglianin could provide the necessary signal to achieve proper scaling of the two tissues, which is necessary for proper flight. Now with that, I would like to thank my advisor, the lab, and the thesis committee for their tremendous support, the funding sources, and the Stock Center for their reagents. Thank you for listening.
That was really interesting. I had a couple of questions. In the glial cell myoglianin knockdown, is there an effect on brain size or on size of the imaginal discs?
Yes, correct. So myoglianin has a role in proliferation of the neuroblasts in the brain, so they do have smaller brains.
And in the whole animal mutants, is there an effect on overall larval size?
It's slight, but it's not as severe as what you see in the wing disc. So the overall body is smaller by maybe 5–10%. We did also measure the muscle size because we thought it might be working like the vertebrate myostatin, You could see overgrowth of the muscle, but we did not see that. Yes.
If I understood correctly, you expect there to be just kind of a tonic phosphorylation of SMAD proteins through the baboon receptor. Is that right?
Correct. You would expect that.
And is that, I mean, I thought when people did phospho-SMAD stains, they saw this very nice gradient without, kind of a flat basal level, which is, I guess, what your hypothesis would say, that there's constant signaling during imaginal disc growth from this muscle signal. Is that my understanding the model?
So phospho PMAD, you mean, or versus phospho DSMAD2?
Yeah, I guess I had thought that the antibody recognized both, but I'm not. This isn't my field.
No,
so.
it does not recognize both.
Okay.
Yes.
Hello. I'm quite curious why you finally chose to research or focus on myoglobin.
That's just where the DSMAD2 phenotype led us to follow that path. I mean, it was just due to mapping of the pathway that led us to track down myoglobin.
Okay, and the follow-up question is, when you lock down myoglobin in skeletal muscle cells...
Sorry, we have to move on to the next talk. Thank you, Ambuj.